Sunrise spills across a Colorado dairy, painting metal roofs gold as rows of black and white Holsteins shuffle toward the milking parlor. The scene feels timeless, wholesome, even nostalgic, until you learn that several of these cows have tested positive for H5N1, a strain of bird flu once thought chained to barns filled with chickens and turkeys. The virus has made an audacious leap across species, trading feathers for fur and placing breakfast tables on alert. This chapter sets the stakes: how a microbe forged in a poultry shed now lurks in the very milk most families pour without a thought. For decades, H5N1 haunted headlines whenever it burned through Asian poultry markets, but infections in mammals were rare and often dead-end. That changed in late March 2025, when veterinary labs in Texas and Kansas isolated the virus from dairy cows suffering sudden drops in milk yield, fever and sluggishness. Genomic sequencing revealed a handful of mutations in the receptor-binding pocket of the hemagglutinin protein, tweaks that let the virus grasp bovine respiratory cells with unsettling ease. Within 2 weeks, state officials counted 74 affected herds across 17 counties. The jump stunned epidemiologists. Never before had avian influenza found a comfy foothold in cattle. Milk trucks kept rolling, yet whispers spread at breakfast bars and grocery aisles. Could the virus flow from udder to carton? Raw milk advocates insisted pasteurization would neutralize any threat, but early digital rumors traveled faster than lab confirmations. Social feeds exploded, with cell phone footage of farmers dumping milk into trenches, blue plastic gloves piled nearby. Although the Food and Drug Administration quickly stated that commercial pasteurization inactivates H5N1, trust drained as quickly as spilled milk. The emotional fracture ran deeper than commerce. Dairy farms are family farms. Cows have names, personalities, and a daily rhythm entwined with workers who see them more than their own kids. A virus crossing the barn door felt like betrayal by nature itself. Farmers who once feared price swings now worried about biosecurity inspections, quarantine tape, and culling orders. Meanwhile, consumer parents toggled between sympathy and self-preservation, scanning cartons for origins, Googling safe milk temperature, and quietly shifting to oat lattes. Our journey starts at this point of tension. The leap from bird to cow is both biological and symbolic, a warning that ecological boundaries are more porous than we like to admit. Over the next 5 chapters, we will track the virus from its avian cradle through cattle corridors, into economic spreadsheets and ethical debates, and finally toward the collective choices that can stop the next spillover before it starts. Milk marketing boards quickly convened crisis panels, crafting messaging that walked a tightrope between reassurance and transparency. Television commercials depicted smiling nutritionists pouring glasses of milk next to animated shield icons labeled pasteurized protection. Meanwhile, farmers live-streamed daily routines on social platforms, fielding questions about tractor disinfection and calf immunity. This grassroots storytelling forged unexpected alliances. Urban food influencers partnered with rural farmers. Veterinary behaviorists observed subtle stress indicators. Tail flick frequency reduced rumination that correlated with subclinical infection. Incorporating these biomarkers into barn management apps turned smartphones into sentinel devices. Every data point became a pixel in a rapidly updating heat map, illustrating the outbreak in living color, far more vividly than any press release. By the chapter's end, one statistic crystallizes urgency. In just 6 weeks, the virus traveled from a single index herd to facilities across 9 states, with an estimated 110,000 cows at risk of exposure. This number reframes the issue from a curious anomaly to a national biosecurity litmus test. Within this maelstrom, culinary culture reacted in surprising ways. Farm-to-table restaurants replaced seasonal cheese plates with creative plant-based spreads, explaining sourcing decisions on menu footnotes that doubled as mini public health PSAs. Food bloggers filmed side-by-side taste tests between traditional burrata and almond ricotta, framing the shift not as deprivation but as chef-driven innovation. These gastronomic adaptations illustrate a broader principle when biology redraws the menu, culture. How exactly did a virus tailored for warm-blooded dinosaurs break into the world of cud-chewing ruminants? Investigators pieced together a mosaic of plausible paths. Migratory snow geese, shedding the virus along their transcontinental flyways, grazed in corn stubble that double-functioned as winter forage for free-range cattle. Spillover likely began there, on shared ground soaked with droppings invisible to the hurried eye. Once inside a bovine, the virus found respiratory tissues cooler than avian air sacs, yet surprisingly permissive, giving it time to mutate for better fit. How exactly did a virus tailored for warm-blooded dinosaurs break into the world of cud-chewing ruminants? Investigators pieced together a mosaic of plausible paths. Migratory snow geese, shedding the virus along their transcontinental flyways, grazed in corn stubble that double-functioned as winter forage for free-range cattle. Spillover likely began there, on shared ground soaked with droppings invisible to the hurried eye. Once inside a bovine, the virus found respiratory tissues cooler than avian air sacs, yet surprisingly permissive, giving it time to mutate for better fit. Geneticists traced 3 key amino acid substitutions that increased affinity for alpha-2, 6-sialic acid receptors prevalent in mammals. One change, T192I, had appeared before in ferret experiments, but vanished in nature, until now. Coupled with deletions in the neuraminidase stalk that favor mucus penetration, the virus gained a passport to bovine lungs. Crucially, cattle shed virus in nasal secretions far longer than infected chickens, offering a wider transmission window among tightly packed milking lines. Surveillance struggled to keep pace. Dairy herds undergo routine testing for brucellosis and bovine tuberculosis, but influenza swabs were alien to most barns. Early cases masqueraded as mastitis or respiratory syncytial virus. Only when farm workers reported clusters of flu-like illness among themselves did public health labs widen their scope. Fortunately, no human infections proved severe, but each negative result felt like a coin flip in an escalating roulette. Biosecurity audits revealed simple breaches. Shared equipment between poultry sheds and dairies, trucks rolling from chicken litter piles to feed silage without disinfection, and farm dogs chasing ducks through open gates. The virus did not need wings once human routine paved the way. All viruses mutate, yet not all manage to rewrite the ecology of 2 industries overnight. H5N1 accomplished this feat with the help of our globalized food system, long supply chains, just-in-time feed deliveries, and the modern appetite for cheap protein. In doing so, it reinforced a sobering truth: biosecurity is only as strong as the weakest daily habit on the farm. As we proceed, remember that every spillover story is also a tale of human shortcuts and structural incentives that invite microbes to explore new hosts. Epidemiological interviews revealed that seasonal labor patterns unintentionally bridged poultry and dairy sectors. Workers rotated between turkey processing plants in winter and dairy milking lines in spring, carrying boots still crusted with litter. Language barriers and lack of paid sick leave compounded risks. Symptomatic employees often powered through shifts to avoid lost wages. Public health outreach campaigns scrambled to create multilingual videos explaining testing protocols and wage relief grants. Transport logistics emerged as silent accelerators. In many regions, milk tanker drivers handle hoses and fittings at dozens of farms per route. A single contaminated gasket reused without sanitation could seed multiple facilities before noon. Recognizing this, a coalition of driver unions and processors piloted single-use couplers embedded with tear-away sterilization seals. Early data suggested a 60% reduction in cross-farm contamination incidents. Perhaps most mind-bending was the discovery of avian influenza RNA in farm dust samples collected from soybean fields adjacent to infected dairies. Wind-borne particles swirling invisibly blurred the line between barn and wild ecosystem. Atmospheric scientists joined virologists to model plume dispersal, emphasizing that spillover containment must consider not just roads and rivers, but air currents that respect no fence line. By mid-April, federal agencies raced to build a defensive maze around infected dairies. Checkpoints monitored cattle transport, milk tankers required bleaching protocols, and veterinarians launched statewide surveillance. Yet the virus threaded the gaps. Real-time PCR kits backordered as global demand spiked, leaving some farms waiting 10 days for confirmation. In that limbo, asymptomatic cows kept moving through regional auction yards. By mid-April, federal agencies raced to build a defensive maze around infected dairies. Checkpoints monitored cattle transport, milk tankers required bleaching protocols, and veterinarians launched statewide surveillance. Yet the virus threaded the gaps. Real-time PCR kits backordered as global demand spiked, leaving some farms waiting 10 days for confirmation. In that limbo, asymptomatic cows kept moving through regional auction yards. Newsrooms scrambled for footage, but many clips featured serene, spotless parlors. Advanced camera filters masked the exhaustion of workers scrubbing every surface with virucidal foam. Algorithms on social media platforms prioritized bright thumbnails over gritty realism, inadvertently downplaying the crisis. Midway through our story, it is vital to pause and invite the audience into action. Share reputable updates, challenge sanitized imagery, and subscribe to EcoEra so we can keep following the evidence wherever it leads. Testing weaknesses were compounded by policy friction. Unlike poultry, where culling entire flocks is standard, dairy cows represent individual capital assets worth thousands. A federal indemnity program existed, but calculations lagged behind market fluctuations. Some producers hesitated to report suspect cases, fearing financial ruin more than the pathogen itself. When epidemiologists plotted transmission chains, silent nodes consistently aligned with farms delaying disclosure by even 48 hours. At the consumer end, pasteurization remained the bedrock of safety. Yet the phrase "remains effective" sounded less comforting amid endless push notifications. A small but vocal raw milk movement framed the outbreak as a conspiracy against natural food, urging followers to support local and trust gut immunity. These messages traveled unfiltered across platforms, reminding us that misinformation evolves as rapidly as any virus. If biosecurity lapses gave H5N1 its opening, communication lapses flung the doors wide. Crisis fatigue, algorithmic bias, and economic incentives synchronized like gears in a flawed clock, marking time for the virus to explore new pastures. We now follow its ripple effects beyond the farm gate. Although laboratories and extension services disseminated emergency protocols, adoption varied by region. In states where consolidated mega-dairies dominate, corporate safety officers issued standardized checklists and digital tracking badges for staff. Smaller family farms, often operating on razor-thin margins, relied on handwritten logs and homemade disinfectant stations cobbled together from garden sprayers and PVC pipe. These disparities mapped neatly onto subsequent case clusters, drawing a stark line between those who could afford preparedness and those who could not. Satellite data confirmed what on-ground reports hinted. Truck movement between high-incidence counties continued at nearly pre-outbreak levels, a testament to the economic gravity pulling fresh milk toward processors no matter the risk. Epidemiologists borrowed network theory from social media analytics to model tanker routes, identifying supernodes where one contaminated valve could theoretically seed dozens of facilities. The exercise illustrated a brutal feedback loop. The same distribution efficiency that keeps grocery shelves stocked can also accelerate pathogen spread when safeguards slip. A deeper dive into communication breakdowns uncovered algorithmic culprits. Social media platforms, tuned to boost engagement, inadvertently prioritized comforting content over alarming yet accurate reports. Posts featuring pastoral cow imagery paired with reassuring captions outperformed lab updates in the algorithmic lottery, creating a perception gap. Behavioral economists coined the term pastoral filter bubble to describe this soothing distortion. Recognizing the stakes, a coalition of fact-check or markets react faster than microbes. Futures for Class 3 milk used in cheese production fell 9% the week the outbreak became public, wiping out millions in projected revenue. Export partners demanded origin certificates for every shipment, forcing co-ops to scramble for paperwork. Small artisanal creameries, once cushioned by loyal clientele, confronted price spikes in veterinary care, cleaning supplies, and insurance premiums. Meanwhile, supermarket chains hedged by ordering extra shelf-stable, plant-based alternatives, accelerating a consumer pivot that had long simmered under the surface. Markets react faster than microbes. Futures for Class 3 milk used in cheese production fell 9% the week the outbreak became public, wiping out millions in projected revenue. Export partners demanded origin certificates for every shipment, forcing co-ops to scramble for paperwork. Small artisanal creameries, once cushioned by loyal clientele, confronted price spikes in veterinary care, cleaning supplies, and insurance premiums. Meanwhile, supermarket chains hedged by ordering extra shelf-stable, plant-based alternatives, accelerating a consumer pivot that had long simmered under the surface. Yet the ledger shows intangibles beside dollars. Farm workers worried not only about infection but also about bringing the virus home to immunocompromised relatives. Veterinarians faced the moral weight of recommending culling beloved bovine companions. Public trust in regulatory agencies wavered, rekindling memories of past foodborne outbreaks. Each sector wrestled with its own definition of acceptable risk. Ethicists joined the debate, asking whether our drive for high-density, high-yield animal operations had created a perfect evolutionary workshop for pathogens. They pointed to European surveys suggesting that farms with pasture access reported lower viral loads, hinting that welfare and public health intertwine. The industry countered with data showing controlled indoor environments reduce exposure to wild birds, illustrating that solutions rarely fit into neat ideological boxes. One underreported casualty was the veterinary student pipeline. Applications to large animal programs, already in decline, took another hit as headlines portrayed barnyards as biohazard zones. The next generation of experts may hesitate to enter a field haunted by invisible threats. Still, crises can catalyze reform. Dairy cooperatives in Wisconsin piloted pooled biosecurity funds so neighbors are not financially punished for early reporting. Startups fast-tracked ultraviolet milk filtration systems promising redundancy beyond heat treatment. And a coalition of milk processors and animal welfare NGOs drafted guidelines for transparent outbreak dashboards, so consumers monitor cases like weather reports. A radical experiment in trust through sunlight. Economists at Purdue University projected that if 15% of the national herd became infected, dairy revenue could drop by $4 billion annually, while downstream industries, cheese, butter, infant formula, would feel cascading shortages. But numbers cannot capture cultural loss. In rural county fairs, prize-winning cows symbolize generational knowledge. A cancelled show season drained not just ticket sales, but community identity. Even international tourism took a hit. Farmstay vacations, rising in popularity among urban families seeking pastoral escapes, saw booking cancellations of up to 70%. Local B&B owners scrambled to pivot, offering virtual tours that featured sanitized, pre-recorded footage, another layer of mediated reality that shaved urgency from the crisis narrative. Meanwhile, animal rights groups leveraged the spotlight to question intensive dairying itself, proposing a pivot to plant-based alternatives. The debate, once fringe, entered mainstream talk shows, illustrating how a microscopic organism can become a fulcrum for societal reconsideration of food ethics. Insurance actuaries, faced with modelling an outbreak that bridges avian and bovine domains, introduced a novel metric cross-species contagion risk, CSCR. Policies adjusted nearly overnight, with premiums spiking for farms operating within 5 kilometers of wetlands. This financial nudge mirrored ecological risk maps, subtly steering agricultural expansion away from migratory bird flyways. Whether profit motives can accomplish what regulations have struggled to enforce remains an open experiment. Behind the scenes, laboratories sprinted to decode the pathogen. Within 30 days of the first bovine sequence upload, 3 vaccine candidates entered pre-clinical trials in calves. Using viral-like particles and mRNA platforms, researchers aimed for broad-spectrum immunity that could block both avian and mammalian receptor variants. The challenge lay in economics. Poultry vaccines sell by the million for pennies, but immunizing every dairy cow would cost billions. Behind the scenes, laboratories sprinted to decode the pathogen. Within 30 days of the first bovine sequence upload, 3 vaccine candidates entered preclinical trials in calves. Using viral-like particles and mRNA platforms, researchers aimed for broad-spectrum immunity that could block both avian and mammalian receptor variants. The challenge lay in economics. Poultry vaccines sell by the million for pennies, but immunizing every dairy cow would cost billions. Parallel efforts explored antiviral feed additives, nasal probiotics, and CRISPR-based gene drives in migratory birds, ideas that oscillate between visionary and unsettling. Regulatory agencies balanced urgency with caution, mindful that a misstep could seed new ecological dilemmas. Yet collaboration blossomed. Geneticists in Kyoto shared structural models with immunologists in Ontario via open-source repositories, compressing discovery timelines. Field studies measured viral persistence in raw milk, finding infected particles survive up to 8 hours at refrigeration temperature, but perish within 15 seconds during standard pasteurization. This data fortified public messages that dairy remains safe, though risk perception rarely bends instantly to charts. Perhaps the most quietly heroic work happened in extension offices, where specialists taught farmers to build disinfectant footbaths, adjust airflow, and establish animal quarantine pens. Success stories multiplied. Farms that implemented rigorous entry protocols saw secondary infection rates plummet by 80%. These victories seldom make the evening news, but they tip the balance from inevitability to control. Science shines brightest when translated into daily practice. Each incremental improvement, a redesigned sanitizer nozzle, a faster PCR cycle, a crowdsourced genomic dashboard, builds the invisible levee holding back the storm. Yet no levee endures forever without collective maintenance. The ingenuity race extended beyond vaccines. Engineers in Finland trialed a rapid aerosol disinfection system for enclosed barns, utilizing hydrogen peroxide mist deployed by autonomous drones. Preliminary results showed a 98% reduction in airborne viral particles after a 3-minute cycle. Elsewhere, data scientists developed machine learning algorithms that listen to barn acoustics. Subtle changes in cow coughing patterns predicted infection 2 days before clinical signs, providing precious lead time for isolation measures. Universities partnered with indigenous knowledge holders to integrate traditional land stewardship practices, rotational grazing, diversified pastures, wetlands restoration, to reduce wildlife-livestock interface. These ecological interventions, often sidelined in high-tech conversations, demonstrated measurable decreases in wild bird congregation near feedlots. The message was clear. High-tech and deep tradition are not adversaries, but complementary pillars in a resilient biosecurity architecture. Public interest in these breakthroughs generated unprecedented crowdfunding campaigns. A global platform raised $10 million in a week to subsidize diagnostic kits for low-income farms, democratizing access to early detection. It was a vivid reminder that science thrives when citizens invest not only attention, but tangible support. Birds, cows, people, 3 classes of vertebrates sharing one viral breath. The H5N1 dairy outbreak is a parable of interconnectedness. Climate-driven migration shifts birds into new flight corridors. Agricultural systems place different species elbow to elbow. Smartphones stitch humans into instant networks of information. True or misleading, every boundary we draw, microbes rewrite in aerosol. Birds, cows, people, 3 classes of vertebrates sharing one viral breath. The H5N1 dairy outbreak is a parable of interconnectedness. Climate-driven migration shifts birds into new flight corridors. Agricultural systems place different species elbow to elbow. Smartphones stitch humans into instant networks of information. True or misleading, every boundary we draw, microbes rewrite in aerosol. Looking forward, policymakers debate a One Health surveillance network linking wildlife rangers, veterinarians, and hospital labs in real time. Funding remains uncertain, but the concept gains traction. Monitor the ecosystem like a cardiogram, treating anomalies before they cascade. Meanwhile, consumer choices, buying pasteurized dairy, supporting farms with robust welfare and biosecurity demanding transparency, signal market forces as potent as any vaccine. For farmers who watched beloved herds fall ill, recovery will be measured not just in herd size, but in renewed community resilience. For scientists, the outbreak underscores the value of open data and global cooperation. For viewers, it reframes a morning ritual. Each sip of milk carries a story of biology, technology, and vigilant stewardship. If this journey through barns and boardrooms has stirred your curiosity and resolve, share the episode, join our growing community, and keep demanding unvarnished truth in the swirl of digital noise. Because microbes never sleep, and neither should our commitment to understanding them. Together, we can ensure that the next mutation meets a world prepared, not panicked. Policy discussions reached the United Nations Food and Agriculture Organization, where delegates debated a voluntary treaty on agricultural spillover prevention. Proposals included standardized reporting portals, a global fund for rapid response, and technology transfer clauses ensuring lower-income nations can access advanced diagnostics. While negotiations trudged through diplomatic labyrinths, NGOs forged ahead with regional pilot programs linking displaced poultry workers, dairy cooperatives, and conservationists in a shared surveillance grid. At the personal level, school curricula adapted, weaving zoonotic disease lessons into biology classes. Students role-played as epidemiologists, tracing imaginary outbreaks on classroom maps, learning the gamut from PCR to public speaking. This generational education may be the most potent vaccine of all, an inoculation against ignorance. Yet fatigue lurks, news cycles pivot quickly, and the temptation to declare victory arrives long before the virus retreats. Each viewer carries a part of the solution. Resist the lull, stay informed, demand accountability from supply chains. When you lift a glass of milk tomorrow morning, consider the invisible web that delivered it. Migratory birds, farmhands, truck drivers, lab technicians, policy drafters, and yes, the microbes forever testing our vigilance. No single action ends the threat, but collective, sustained effort bends its trajectory. Let the legacy of the H5N1 dairy outbreak be a turning point where science, industry, and society learn to breathe in sync, guarding the shared air that binds us. Weeks after the outbreak's first anniversary, a multidisciplinary symposium in Denver convened veterinarians, virologists, ethicists, economists, and storytellers. Their unanimous conclusion, the memo of 2025, is that microbial threats are narrative threats. How societies tell these stories, who is framed as protagonist, who bears blame, what futures are imagined, shapes behavior as deeply as any viral replication cycle. Eco-Era commits to holding space for those narratives, amplifying voices that too often As the credits on this chapter roll in your mind, remember that surveillance technology, vaccination pipelines, and cross-sector treaties are only as strong as public will. Clicking away unchanged is the quiet mutation complacency craves. The alternative, staying curious, asking hard questions, turning knowledge into action, writes a different evolutionary story, one where microbes still surprise us, but never again catch us asleep at